mAbxience is an international biotechnology company specializing in the research, development, manufacture and global commercialization of biosimilar medicines, primarily monoclonal antibodies. A biosimilar is a biological medicinal product that is equivalent in quality, efficacy and safety to the reference biological medicinal product. The similarity exercise ensures these attributes using state-of-the-art and highly sensitive analytical methods that determine the physicochemical structure and functional properties of the biosimilar as compared to the reference medicinal product. Mass spectrometry is used to determine the primary structure, that is, the amino acid sequence. The molecular weight is determined by intact mass analysis. The amino acid sequence is determined by peptide mapping, which is based on protein digestion and peptide identification. The molecular conformation is then studied. Disulfide bonds analysis demonstrates the conformation of the biosimilar as compared to the reference molecule, which is essential for biological activity. Chromatographic techniques are used to determine post-translational modifications, that is, modifications after protein synthesis, by analyzing acidic and basic variants and the N-glycan profile. This is the most important modification, affecting activity and immunogenicity. Purity is analyzed using specific techniques, such as molecular exclusion. This technique allows the molecule to be separated from its aggregates and other fragments based on molecular size. Finally, the biological activity is assessed. This is carried out in vitro using specific assays and in vivo to characterize the mechanisms of action. As the innovator, the biosimilar can prevent non-specific binding of growth factors to cells, thus inhibiting uncontrolled cell proliferation. This comprehensive exercise enables mAbxience to ensure the similarity of the biosimilar to the reference product. This, together with confirmation in preclinical and clinical trials, will allow the biosimilar to be approved and subsequently commercialized. At mAbxience, we develop, manufacture and market biosimilars, mainly monoclonal antibodies, using the most advanced technology and cutting-edge analytical methods in our processes, following strict international regulatory guidelines. First, an in-depth analysis of the reference product is carried out to obtain all the possible information about its structure. This is a stage in which a considerable amount of time is invested, guaranteeing the best results. In the transfection phase, once the DNA sequence that corresponds to the antibody is synthesized, it is inserted into the plasmid. The plasmid with the DNA transfects the cell, where the gene corresponding to the antibody and other genes needed for its translation are integrated randomly into the cell genome and thereby producing the desired antibody. In this stage, called random insertion, a set of polyclonal cells is obtained. These cells produce different quantities of antibodies with minor differences. The development of the cell line phase starts. During this phase, the cells are isolated and analyzed using different techniques until only the one that produces the most similar antibody to the innovator in terms of quality remains. This is when it is frozen and the cell bank is obtained. This is the basis for the following phase, upstream process. In this phase, the vial is thawed and cultivated, first in flasks by shaking, before being put into the bioreactors. The technology used is single-use, which provides greater flexibility for production and prevents cross-contamination via the use of disposable materials. In the bioreactors, nutrients and physicochemical parameters are vital for cell growth. As such, temperature, pH or oxygenation are strictly controlled during this phase. In the bioreactors after the production phase, all remaining impurities are eliminated in the purification or downstream process using chromatography columns and filters, which, by means of different resins, enable all the undesired components to be discarded whilst retaining only the antibody which is the most similar to the reference product. Lastly, during the formulation phase, and before filling the vials or syringes with the final product, the antibody is added to a solution containing the necessary excipients to stabilize it and make it suitable for patients. This is how mAbxience develops and manufactures biosimilars, with focus on quality throughout the whole process, to achieve that each product delivered to patients is highly similar to the reference product. We call this fingerprint-like biosimilars by design.